Welcome back, everybody. Today, we're going to take a deep dive into something that's pretty amazing, a brand new AI model called Evo2.
Evo2.
And what makes this so cool? Well, imagine a computer program that not only reads the entire genetic code of millions of organisms,
Right.
but also understands it so well that it can actually predict how changes to that code will affect everything,
Yeah.
from individual molecules to entire organisms.
It's really remarkable.
It really is.
Yeah, it's like having a virtual geneticist right at our fingertips, capable of unraveling the complexities of DNA in a way that we've never seen before.
Okay, so let's unpack this a little bit.
Okay.
Evo2 has been trained on an enormous amount of DNA and RNA data.
Uh-huh.
We're talking trillions of base pairs.
Massive data.
So it's got this incredible grasp of the language of genetics, but it's not just memorizing sequences.
Right.
It's actually learning the underlying rules and logic, like figuring out where genes start and stop.
Yeah.
Those crucial signals that tell the cell's machinery where to begin and end reading the instructions to build a protein.
And that's what's so fascinating, is that Evo2 was able to identify these start and stop signals, even though that information wasn't explicitly pointed out in its training data.
Yeah.
It's as if it learned the grammar and punctuation of the genetic code just by analyzing the vast amounts of data it was given.
So it's almost like teaching a child to read by giving them a library full of books.
That's a great analogy.
Then letting them figure it out on their own.
And just like a child who learns to read can then start writing their own stories, Evo2 can apply its understanding of genetic patterns to make predictions about things it's never encountered before. And the researchers call this zero-shot learning.
Okay, so this is where it gets really interesting, right? Because once you understand the rules, you can start playing with them.
Yep.
And that's what Evo2 does. It can predict how changes to the genetic code or mutations will affect the function of a gene or even an entire organism. Exactly.
They call this predicting the fitness of a sequence.
And think about the implications of this.
Yeah.
Imagine being able to predict how a specific mutation in a patient's DNA might increase their risk for a certain disease.
Oh, wow.
Or how a particular genetic variant might affect their response to a drug.
That's personalized medicine on a whole new level. So it's not just about understanding our genetic makeup. It's about predicting how those genes will actually function.
Exactly.
And how that will affect our health and well-being.
Precisely. And they've tested Evo2's prediction capabilities using various techniques.
Okay.
One of them is called deep mutational scanning, which is basically a way to experimentally measure the effects of mutations on a protein.
Yeah.
And what's remarkable is that Evo2's predictions often match or even outperform the accuracy of methods that rely on tons of experimental data.
So it's almost like skipping the lab work.
Yeah.
Getting those insights directly from the genetic code itself.
Exactly.
That's a game changer for research.
Yeah.
But let's not forget about RNA.
Remember how I mentioned Evo2 was trained on both DNA and RNA data? Well, this means it can also analyze RNA sequences, which play a crucial role in how genes are expressed.
Right. And one of the things they discovered is that Evo2 can actually predict the stability of human messenger RNA or mRNA molecules just by analyzing their sequences.
Now, for those who might not be familiar with mRNA, it's like the messenger that carries genetic instructions from DNA to the ribosomes, which are the protein-making factories of the cell.
Exactly.
And the stability of mRNA, how long it sticks around, affects how much of a particular protein gets made.
Exactly. So if Evo2 can predict mRNA stability, it can give you a glimpse into how active a gene is likely to be.
Right.
And they found that Evo2's predictions lined up incredibly well with experimental data on mRNA decay rates.
That's pretty mind-blowing. It's like Evo2 is giving us a sneak peek into the inner workings of our cells.
Yeah.
Showing us which genes are likely to be turned up or down based on the stability of their mRNA messengers. But wait, there's more. It turns out Evo2 can also tackle human clinical data sets like ClinVar and BRCA1, which contain information about genetic variants and their links to diseases.
Yes, and this is where things get really exciting in terms of potential applications.
Yeah.
Evo2 did particularly well in predicting the effects of variants in the non-coding regions of DNA, which have traditionally been very difficult to analyze.
So we're talking about those vast stretches of DNA that don't directly code for proteins, but can still have a huge impact on how genes are regulated.
Exactly.
And whether someone might be predisposed to certain diseases.
Right.
And Evo2 is cracking the code, so to speak.
That's the amazing part. For a long time, the focus has been on protein-coding genes.
Yeah.
But the vast majority of our DNA is non-coding.
Right.
And it's becoming increasingly clear that these non-coding regions play a crucial role in gene expression and can contribute to disease.
Yeah.
So being able to analyze them effectively is a major breakthrough.
It's like we're finally starting to see the whole picture, realizing that there's so much more to our genetic story than just the genes that make proteins.
Exactly.
But how did the researchers figure out what was going on inside Evo2? It's almost like it's thinking right.
Well, to understand what kinds of features Evo2 was learning, they used a technique called sparse autoencoders.
Okay.
Think of it like this. They designed a special tool to peek inside Evo2's brain and see what it was learning.
And what did they find?
What they discovered was astonishing. Evo2 was picking up on all sorts of biologically meaningful patterns, even though it wasn't explicitly told to look for them.
Wow.
For example, it could distinguish between the parts of a gene that code for proteins called exons and the non-coding parts called introns.
Right.
It could also identify those regions where proteins bind to DNA to regulate gene expression.
So it's not just memorizing sequences, it's actually learning the intricate dance of molecules, the complex interactions that govern how genes are turned on or off,
Yes.
how they're expressed, and how they ultimately shape who we are.
Exactly. And to really put Evo2 to the test, they gave it a challenge that sounds straight out of a science fiction movie. They used it to analyze part of the woolly mammoth genome.
Oh, wow.
And it accurately annotated the mammoth's genes,
Right.
showing its potential to help us understand even extinct species.
Now, that's what I call taking a deep dive into the past.
Right.
But wait, there's something even more futuristic that we need to talk about. Evo2's ability to not only read DNA, but to actually generate entirely new DNA sequences.
Yes, you're right. We need to discuss that.
Okay.
They tested this ability by having Evo2 generate mitochondrial DNA. Mitochondria, as you know, are those tiny powerhouses inside our cells that have their own separate DNA.
Right, they're like little mini genomes within our cells.
And what's amazing is that Evo2 was able to create plausible mitochondrial genomes that contained all the expected genes in the right order.
Hold on, so it's not just reading and understanding DNA, it's actually writing new genetic code. That's like stepping into the shoes of a creator, isn't it?
It does blur the lines, doesn't it? And it didn't stop there. They even had Evo2 generate a complete genome for a type of bacteria with a very small genome called Mycoplasma genitalium.
Wow, that's almost stepping into the realm of science fiction. But before we get too carried away, we need to take a step back and ask ourselves, what are the implications of this? What does it mean when AI can not only read, but also write the code of life?
That is the million-dollar question, isn't it?
Got it.
And it's one that we'll delve into deeper in the next part of our deep dive.
Welcome back. Last time we left off on this amazing note about Evo2's ability to generate entirely new genomes.
Right.
Basically writing new genetic code. It's almost like stepping into the shoes of a creator.
It's a remarkable feat, no doubt. It raises so many fascinating questions about the nature of life itself and our ability to understand and even manipulate it.
Okay, so let's get down to the nitty-gritty. How do they actually test these generated genomes?
Right.
How do they know if Evo2 is creating something plausible or just a jumble of genetic gibberish?
Well, they use a combination of clever tools and techniques to analyze and evaluate the generated sequences.
Okay.
For example, when they had Evo2 generate a complete genome for that bacteria we talked about, Mycoplasma genitalium, they used a tool called Prodigal to identify potential protein-coding genes within the generated sequence. It's kind of like a genetic spell checker, looking for patterns that indicate where a gene might be located.
Interesting. So they're making sure that these new genomes actually contain genes that could potentially make proteins,
Right.
those workhorses of the cell. But how do they know if those genes are plausible, if they resemble anything we've seen in nature?
Right. That's a crucial step. They use a massive database called FAM, which contains information about protein families and domains,
Okay.
those functional units within proteins. They compare the predicted genes from the generated genome to the known genes in FAM to see if there are any similarities in the evolutionary relationships.
So it's like a genetic family tree, tracing the ancestry of these new genes to see if they have any known relatives.
Right.
And what did they find?
They found that a significant portion of the genes generated by Evo2 actually had recognizable counterparts in the FAM database.
Wow.
Indicating that they weren't just random sequences, but potentially functional genes.
Okay.
But they didn't stop there.
Yeah.
To further assess the plausibility of these genes, they used another tool called HHPRED.
HPRED.
This one compares protein sequences to find similarities and evolutionary relationships.
So they're really putting these new genes through the ringer, making sure they hold up to scrutiny.
Yeah.
It's not enough just to have genes. They need to be genes that could potentially work in a real biological system.
Exactly.
But genes are just instructions. What about the actual proteins they code for?
That's where things get even more interesting. They took those predicted proteins and used a tool called ESM-Fold to predict their three-dimensional structures. You see, the shape of a protein determines its function. It needs to fold up in just the right way to interact with other molecules and do its job.
Ah, so it's like a key fitting into a lock.
Exactly.
The shape has to be precise for the protein to work properly.
Exactly. And when they analyzed the predicted structures of the proteins encoded by Evo2's generated genes,
Okay.
they found that many of them resembled known protein folds and interactions, suggesting that they could potentially function in a real biological system.
That's incredible. So Evo2 isn't just generating random genetic sequences.
Right.
It's actually designing blueprints for the building blocks of life.
It's a powerful way to think about it. But of course, all of this is still in the digital realm.
Right.
Actually synthesizing these genomes, getting them into cells, and seeing if they truly function, those are significant hurdles yet to be overcome.
Right, of course. But still, it's mind-boggling to think that we're at a point where AI can design plausible genomes. What other amazing things can Evo2 do with this generative power?
Well, they also explored its potential in the field of epigenomics.
Epigenomics. Okay, that's one of those words that makes my head spin a little. Can you break it down for me?
Think of it this way. If the DNA sequence is the hardware of the genome genome is the software. It's a layer of information on top of the DNA that controls which genes are turned on or off in different cells and at different times.
Ah, so it's like the control panel.
Exactly.
That fine-tunes how the genetic code gets expressed, making sure the right genes are active at the right time.
Precisely. And it's incredibly complex, involving chemical modifications to the DNA and the proteins that package it. What the researchers did was try to guide Evo2 to generate DNA sequences that would lead to specific epigenetic patterns.
So they weren't just generating random genomes. They were trying to design genomes that would have specific effects on how genes are regulated. That's taking it to a whole new level of control.
Exactly. They used two other AI models, Informer and Borzoi, which are really good at predicting chromatin accessibility. That's a measure of how open or closed the DNA is, which influences gene expression. They use these models as a feedback system to guide Evo2's generation process.
So Evo2 generates a sequence, Informer, and Borzois analyze it to see what its predicted epigenetic effects would be. And then Evo2 adjusts its design based on that feedback. It's like a genetic feedback loop.
That's a great analogy. And the results were impressive. They designed different peak patterns, essentially regions of open and closed chromatin.
Right.
And Evo2 was able to generate sequences that match those patterns surprisingly well.
So they could essentially dial in the desired level of genome activity in different parts of the genome.
Yeah.
That's incredible.
It is. And they found that the more computing power they used during the generation process, the more precise the epigenetic control became. It highlights the incredible potential of this approach to design sophisticated genetic systems with fine-tuned control over gene expression.
The possibilities are mind-boggling.
I know.
Imagine being able to design cells that produce specific proteins on demand, engineer tissues for transplantation, or even create new biomaterials with tailored properties.
It truly is a revolutionary moment in biotechnology. But of course, as with any powerful technology, we must proceed with caution and consider the implications of wielding such control over the building blocks of life.
You're right. It's not just about the can we, but also the should we. And that brings us to the next part of our deep dive, where we'll explore some of the broader implications of Evo2 and its potential impact on society. Welcome back to the deep dive. We've been exploring this groundbreaking AI model called Evo2, which is not only changing how we understand DNA, but also giving us the ability to potentially design new genomes, essentially writing new genetic code. It's mind-blowing stuff, but it also raises some big questions about how this technology should be used.
Absolutely. It's like we've been handed this incredibly powerful tool, and now we need to figure out how to use it responsibly.
So let's talk about those big questions. What are some of the potential challenges or concerns that come with having this level of control over the building blocks of life?
Well, one of the key challenges is predicting the unintended consequences of our actions. Even with the best intentions, introducing new organisms or genetic modifications into ecosystems could have unforeseen and potentially harmful effects.
It makes you think of that saying, life finds a way. We might think we have everything under control,
Right.
but nature has a way of surprising us. It's a bit like opening Pandora's box, isn't it?
It is a complex issue. We need to proceed with caution conduct, thorough risk assessments,
Yeah.
and consider the potential impact on the delicate balance of ecosystems.
Okay, that makes sense. But what about the potential for misuse? Could this technology fall into the wrong hands and be used for harmful purposes?
That's certainly a concern that needs to be addressed. Like any powerful technology, Evo2 could be used for purposes that were not intended by its creators. Yeah. It highlights the need for international collaboration and ethical guidelines to ensure that this technology is used for the benefit of humanity and not for harm.
So it's not just about the science itself, it's about having a global conversation about the ethics and responsible use of this technology. It's a big responsibility, but it sounds like the researchers are aware of these challenges.
They are. One of the encouraging things is that they've made Evo2 and its code open source, which means it's accessible to the scientific community. This promotes transparency and allows other researchers to scrutinize and build upon their work, which is crucial for responsible development.
That's good to hear. It's like they're saying, hey, we're all in this together. Let's work together to ensure this technology is used for good.
Exactly. They've also expressed a commitment to engaging with experts in bioethics and working with policymakers to develop guidelines and regulations that promote responsible innovation.
It sounds like they're approaching this with a sense of humility and a recognition that this technology has the potential to reshape our world in profound ways. It's both exciting and a little bit daunting, isn't it?
It is. We're on the cusp of a new era in biology, one where AI is giving us unprecedented power to understand and manipulate the building blocks of life. Yeah. It's a moment of great opportunity, but also one that demands careful consideration and responsible stewardship.
It really makes you wonder, doesn't it? If AI can now read and write the code of life, what does it mean to be human in this new era of biological possibility?
That's a question we'll all be grappling with for years to come. And I have a feeling this is just the beginning of a much larger conversation about the future of life itself.
It's definitely a deep dive that will stay with me for a long time. Thanks for joining us on this incredible journey. Until next time, keep those neurons firing.